Welcome, everybody, to this afternoon's webinar. We're here from RASopathies Network, and our mission is to advance research to improve the quality of life for individuals with RASopathies by bringing together families, clinicians, and scientists. We're delighted that you're here today. As you know, we're all sort of bound together because we have or we love someone who has a RASopathy, which is any one of these syndromes listed here. And these syndromes are caused by dysfunction or malfunction in the genes or the proteins that are part of this pathway that I show here inside of a cell. So we have this molecular link, and we have this phenotypic link, this link of having these syndromes for somebody that we know. So I want to just remind everybody that if we do research on any one of these particular syndromes, that should and could benefit members of the other syndromes because the pathway is activated inappropriately. And so that's how you get some of the overlapping features. We are going to feature Dr. Tamar Green. She's going to tell us about RASopathies and her study of brain and cognitive development in children with Noonan syndrome and NF1. Dr. Green is the assistant professor of psychiatry and behavioral sciences at Stanford University, and she's also principal investigator of the Bridge Lab, which stands for Brain Imaging, Development, and Genetics. And we also welcome her research assistant, Naomi Furman. So I will stop screen sharing now, and you can take over.
So hi, everyone. I'm super excited to be here. I'm going to start with kind of giving giving few moments for Naomi to talk about our research. And I would say that our goal today is mostly to give back. And the way we want to give back today is giving back knowledge that we gain through many people in the community of Noonan syndrome and NF1 and RASopathies contributed to us by coming to our studies and participating. This is a very important way for us to advance the research in RASopathies. So before I start, just a huge thanks to everyone who already came and hoping you guys gonna come in the future. I have nothing to disclose, and Naomi, I'm gonna give you now.
So Dr. Green is going to share a lot more about what we do and the research, but just a quick overview of our lab. I'll just tell you that the Bridge Lab at Stanford is really dedicated to researching the effect of altered genetics on brain development and neuropsychiatric outcomes, with our overall aim of improving children's mental health and development. And we'll go into more details about that. I'll share a link to a website that has this infographic if you're interested in learning more. And here's a glimpse into our team, the team behind the science. But more importantly, and if we can go to the next slide, please. This is, just like Dr. Green said, our opportunity to share results from studies some of you here listening to us have participated in. So first and foremost, I want to take this opportunity to say thank you to everyone who participated and are actively helping us and helped us advance our understanding of RASopathies, which, as you know, are rare, and we're always looking for more participants to join us for studies. So I do want to take a moment to mention that we are currently and actively recruiting participants, children with Noonan syndrome and NF1 for our studies. Happy to answer any questions. All travel costs are covered by Stanford, and there's more to it. So if you're interested or know anyone who's interested and within this age range, we're currently only recruiting in North America, but I urge you to sign up, and I'll share the link in the chat shortly. But again, we thank everyone who already participated and excited to share findings and results from the studies. And back to you, Dr. Green.
Okay, thank you. Two more plugs is that we extended our genetic kind of inclusion. So we currently including any child with or teen with Noonan syndrome without. We're not limiting we're not limited to specific genes that cause the syndrome, and also we expanded our recruitment to Canada. So if you help us spread the word, that would be great. It's very important for future funding to show that the community can come and actually participate in studies. Okay, and after that, I'm going to just dive into the research. Feel free to ask questions during the talk. Beth will monitor them and can ask me. But at the end, we're gonna open to free Q&A. So feel free to do both. What I'm going to cover today is first to explain why we're interested in RASopathies from the scientific point of view, and what kids with the syndrome can teach us about human brain development and also human cognitive development, and why is it an interesting topic to study. Then I'm going to talk a little bit about the connection of RAS-MAPK pathway, the pathways that is affected in RASopathies to other psychiatric conditions like autism. And in the second section of our talk, we're going to cover neuropsychiatric phenotypes in RASopathies with a focus on disorders like ADHD, attention deficit, and autism spectrum disorders. I'm going to show you findings from a research that we conducted in Noonan syndrome. And the most scientifically involved part of this talk going to be the last part that talks about the brain, and I'm going to feature two findings that we recently discovered. So I want to start with this explanation of why RASopathies are of interest in psychiatric research. So the reason for that comes from kind of like flipping upside down the traditional way to conduct research. The traditional way to conduct research in psychiatry will be to collect a group of kids with autism or a group of individuals with schizophrenia, and then look deeper into their diagnosis and learn about the different features and symptoms that they have, and then asking a question whether there's any neural explanation to the symptoms, and then meaning looking at their brain function or structure, and then trying to find a gene that is responsible for that. The challenge in this approach is that there's no one gene that causes schizophrenia or autism, and you end up with a collection of genetic changes that cause this like route of research to be very, very heterogenic and not always very having the robust and replicable results. And the way to flip that, and the way we use it at the lab, is that we start with mutations, known mutation. In our case, we're very interested in mutations that affect the RAS-MAPK pathway. Hence, we are very, very connected to and interested in any RASopathy. And we are then invite you to the lab. What Nomi and I were talking about is the goal is to invite the kids and the families to the lab, learn about their genetics, learn about their brain, learn about symptoms like ADHD and autism, learn about cognitive function, like how a child, and academic function, also how the child reads and how the child is doing math. And then using those to think about the effect of the RASopathies on all these measures, on the behavior, on how a child learns, and what he might be or she might be encounter in terms of psychiatry or behavioral challenges. Now, again, still in background, we're going to talk about the specific genes and a little bit about the science behind it. So as Beth already presented the RAS-MAPK pathway. It's a different presentation of the pathway, but it's the same pathway. My goal here is not to show any specific we don't need to dive too much into that, but I do want to say that the genes in red are genes that are, when they're mutated, they're causing hyperactivation of the RAS-MAPK pathway. And the gene in red, when they're not affected, they're suppressing this pathway. But when they are affected, they're actually stop the suppression. So all of that to say that we do think that a lot of the phenomenon that we see on the brain, on the behavior, are related to activation of the pathway. And that will happen in a lot of the kids with any RASopathy. So we do think that studying RASopathies as a collection of syndrome is a very smart way to learn more and have the more power to detect these effects. Even though that each one of the mutation is relatively rare, together it's not that rare. This section is to emphasize that RASopathies are connected to human cancers, but in a very different way. The RAS-MAPK pathway was studied and discovered because of somatic mutation in the RAS-MAPK pathway. Somatic mutation means that a mutation happens during the course of a person's life in one of their cells, and those being highly connected to malignancies. However, the mutation that cause RASopathies are mutations that happen in germline, meaning that it happens and it entails all the bodies of the cells, but the type of mutation is probably different and milder. But what kind of like the silver lining here is that because a lot of research was done on cancers, now there's thought and thinking about intervention that were invented to treat cancers that were not very aggressive, might have a benefit to children and individuals with RASopathies. Lastly, I want to emphasize that today we're going to talk about Noonan syndrome and neurofibromatosis 1. I'm going to move on. This slide is only to emphasize that Noonan syndrome that we'll talk about today is caused by a collection of genes, but I'm going to talk today a lot about PTPN11, that is the main gene that causes Noonan syndrome, and NF1, that is, as I mentioned, suppressor gene to the syndrome. Both of them will result in activation of the RAS-MAPK pathway. Okay, so as I said, RASopathies are not only connected with physical features, but also with psychiatric and behavioral manifestations. And one of the evidence that the RAS-MAPK pathway as a pathway is connected not only to RASopathies, but probably also to autism spectrum disorder that caused not because of RASopathies is as follows. the Safari gene collection is a collection of genes that being studied and discovered as being highly, in high confidence level related to autism. And what I did here is I took the genes that are on the RAS-MAPK pathway, and I just tested whether either of these genes came or appear in this collection of genes. And as you can see in bold and with an underline is that underlined is that many of the genes in the RAS-MAPK pathway are appearing as high confidence genes in idiopathic autism, meaning autism that is not related to RASopathy. So that's an indication that the pathway is involved or related to developmental effects that are related to issues with social communication, making friends, understanding social interactions. This is kind of like the, and this is again, it's a very complicated pathway, but complicated slide, but what I want to emphasize here is that this in 22, there's been 2 studies that studied a huge population of children with autism. In this case, 45,000, and in this case, 68,000. And in both of them, the RAS-MAPK pathway or genes that encode the RAS-MAPK pathway appeared as new genes that are related to autism. In this case, out of 72 new genes, the SYNGAP, NF1, and PIK3CA came as new genes that related to autism. And in this case, the researchers have identified the RAS-MAPK signaling protein. The protein, when we have gene, it encodes to a protein, and this protein tends to work together. So these collections of protein have been related. It was one of 6 archetype of networks that relate and contribute to autism. So again, it's another evidence that it's very important for not only for individuals with RASopathies, but also to people who are interested in neurodevelopmental conditions to acknowledge and to know that the RAS-MAPK pathway is part of is affecting these conditions. Now I'm switching to the more to the things that we have done, and I hope I have convinced you that the RAS-MAPK pathway is a very important one to study. And even if anyone has like a rare genetic mutation, it actually has much more significance to not only to people with RASopathies, but also to the general population. In this study, we evaluated 40 children with Noonan syndrome and 40 children without Noonan syndrome, and we compare them on a collection of assessments. And I'm going to kind of like go quickly a little bit about the background of Noonan syndrome, so you have context if you are not familiar. So there's many genes that can cause Noonan syndrome, around 13 that discovered today, but 5 of them are the main causes. It happens in 1 to 2,000 individuals. It's the most common RASopathy. The IQ in most children are in the normal range. And we know that 6 out of 10 have a diagnosis of neuropsychiatric disorder in childhood. So what we did in this study, we evaluated 3 domains. The first domain is attention and executive function. I'm going to give 2 examples for what happens when someone has an effect on his attention or executive function. one of the features can be a difficulty to concentrate for more than 5 or 10 minutes in something that is not very engaging. It's not video games, but it's usually connected to something that the child or the teen is not very interested in. This is how we measure attention, and in many of the kids we do find difficulties with that. So this is the domain of the attention, and executive function can be thought about as the ability to plan ahead, to evaluate your work, which we do know that can be affected in RASopathies. The second domain that we evaluated is social skills. What we mostly evaluated is the social communication and ability to to form friendships. So we evaluated that domain. And lastly, we evaluated aggression. And the reason we did that came from the fact that we saw at the beginning, we started to see kids, and most of the parents did complain that there's a lot of can be a lot of aggression expressed that is above and beyond what you expect from a frustration. For example, what does this mean above and beyond? Being said no, or being said no, you cannot do it, creates a behavioral response that is above what you would expect for the frustration that is presented. So now what I'm going to do is I'm going to present the results, and I'm going to walk you through some plots here. So what you see on the x-axis is the different measures that we used. So for example, we measured attention, we measured hyperactivity, we had like a set of other measures that measure executive function. One of them would be how a child can control his emotion. Another measure would be how a child can inhibit. For example, inhibition is needed when you need to wait in turn, or when you really want something but you need to wait until you get it. Another measure we took is planning and organization. The ability to shift from one task to another task is another executive function feature. And the reason those sections are separate from this section is that these were asking the parents about the child, but we also had the ability to test the children who came to us, and we evaluated their attention and their ability to inhibit, but not only by asking the parents, but also by actually measuring it. And the results on the y-axis, what you can see is two things. In red are the results from kids with Noonan syndrome, and in blue are results from kids without Noonan syndrome. And the higher the score in this case, the more affected, or we see more weaknesses or problems. And as you can see, quite across the board, children with Noonan syndrome presented with more weaknesses and more challenges in those domains that I just described. And another thing that I want you to pay attention is that we have this dashed line, and many of the kids were above this dashed line and the reason that I'm mentioning that is this dashed line is a threshold that above it we think, oh, it's actually clinically significant. It's something that we want to address or think about. So it's not only that it's elevated, it was elevated in a way that we thought it's clinically relevant, that need to be addressed. A second, in this section, it's flipped because we are measuring abilities, the ability to inhibit, for example. So we see that for children with Noonan syndrome, they had more difficulties compared to their peers in those sections. And something I want to mention is that we controlled for IQ. It means that we took IQ into account, and those difficulties are beyond difficulties in cognitive function. So we are quite confident they're separated from any challenges that related to pure cognition. So it's another way for us to make sure that those effects are actually happen above and beyond what happened to the cognition. And I'm now going to cover the social skills, and the same rules that I mentioned before. On the x-axis are the measures that we examined, and on the y-axis is how much it affected. And the measures here are different because we're focusing on social skills. So we measure things like the withdrawal is the child would not participate in things, and atypicality is behavior that is a little bit odd or strange or not like what you expect from a child at that age. And we also measure how the child is aware to his surrounding, how much he understands social communication, how he communicates, and how motivated he is or she is to form social interactions. And on this right side, we measure the child himself, and we took two measures. One of them is how well a child recognizes the emotion on a picture that he's being shown. So can he recognize happy or sad, but more nuanced than that, like how well he recognizes facial expression that express affect. And the second measure was a measure of theory of mind that is considered very tightly related to autism. And as you can see again, is that children with Noonan syndrome will score higher or with more difficulties, or lower on the cognitive measures with more weaknesses on almost across the board, and the measures, again, after controlling for the effect of cognition. So I've been showing you that kids with Noonan syndrome compared to control have, as a group, have these more challenges, both in the domain of the attention and in the domain of social skills. Lastly, in this study, we also conducted a formal psychiatric evaluation. As a child psychiatrist, I thought it's important, and the reason I think it's important is also to inform other child psychiatrists of this and provide some documentation, but also for you to take many parents and families. When they go to school, this is the only child with the RASopathy the school encountered, and I thought it's going to be a really important report just to take with you to school. And we have implemented there a very brief summary with points, so I think it's something that is very useful. But to our findings, what we did find is that 34% of our kids presented with oppositional defiant disorder, which means that the child and the parents are experiencing significant difficulties with behavior at home and sometimes at school. We also found high percentage, 35% of children having some autism spectrum syndromes. None of the kids met criteria for the full ASD syndrome, but many of them presented with symptoms. And in the cohort that we studied, that was very young cohort, I didn't mention that before, but it's between 4 and 11, over 60% presented with ADHD symptoms or ADHD disorder. So it means that we see in this population significant difficulties with attention and concentration. I wanted to give a little bit more information about how, because we do not see the textbook autism in kids with Noonan syndrome. So I wanted to describe more in details that the symptoms of autism would be behavioral rigidity, this difficulty to change either from one task to another task, or having difficulty to think outside of what the thought was. It's kind of like inability to be more flexible about it that creates a lot of frustration. Difficulties with transition, as I mentioned, and difficulty in maintaining social relationship. This is simply maintaining friendships with peers. And as I mentioned before, I mentioned that there's a lot of oppositional defined disorder, and we think what's underlying it is that it's heightened irritability. Irritability described like this a constant feeling of being frustrated and without the ability to emotionally regulate this frustration. So this is a feature that we see. I do think that we see it more in Noonan syndrome than in NF1, but we don't have enough testing of that, but that's my impression. Okay, and now I'm gonna do the last part of the talk that talks about brain and the reason why we're interested in the brain, because as I showed you, the kids can encounter a lot of developmental issues, and we do think that what underline those issues happens in the brain, and this is why we're very interested in that, because even if we going to give any medications or any interventions, we would like to measure and see what happens, not only to the behavior, but also to the brain, because this is the organ that actually derives those behaviors. So you can think about it like if someone is coughing, the physician would like to test their lungs and listen to the lungs and maybe take a picture of the lungs. So on the same way, we do want to think about what happens in the brain. Okay, so the way we do that, we have many ways, but I'm going to describe two today. We take pictures of the brain, and of course the brain is not that colorful, but the colors here are just here to explain that we are parcellating, we are dividing the brain to different regions that we know from extensive work in neuroscience that have similar cellular features or serve specific behavioral cognition. So we parcellate the brain, and then we are able to measure different things about these parts of the brain. So for each region, we can measure how the volume of this region, we can measure how the surface area of the brain in that region, meaning if you remember, this brain is inflated, but the brain is very convoluted, so we can measure how much surfaces there is, and we can also measure how thick different regions of the brain, and that's these have tight connection to development as well. And what I'm going to show you today is not related to what we call cortical sections of the brain. This is the cortex of the brain. We also have more deeper structures that called subcortical regions. And those regions are serving different behaviors. I'm going to focus on one. We measure the dorsal striatum. As you can see, the dorsal striatum is a collection of nucleus that are called the cutate and the putamen. And so what you see here in blue and what you see here in pink And those regions are responsible for function selection and planning, for cognitive flexibility to be able to change from one thing to another thing, are responsible to response inhibition. So meaning like, even though I'm compelled to do something, if I'm instructed not to, I'm not doing that. I'm able to inhibit myself. And the reason is obvious, because I already showed you that the kids are struggling with that, and this is a weakness. So I want to look on this nuclei that serve and connected to these regions. So we measure them, as I showed you before, and we measure them in a group of kids with Noonan syndrome. Some of them are caused by PTPN11, and some of them is caused by SOS1, and TD will be our typically developing healthy controls. And what you see on the left is that this is the volume of the structure, and this is the 3 structures that we measured. And as you can see, across these structures, and I'm showing you only the left side, it happens the same on the right side, is that the children with Noonan syndrome have significantly lower volume of these structures compared to typically developing controls. This is the same thing, the same information, but just illustrated on the structure themselves. And as you can see, this is the more blue the color, the smaller the structure, the striatum is. So again, it's the same information, just presented visually different, in a visually different way. And what I want to show here is that I already told you that kids with Noonan syndrome have auditory attention and inhibition weaknesses. As you can see here, the red are lower than the blue bars. And what we did here is we asked whether there's any connection between how big or small your striatum is and how you function. And what we see is that for the kids in the Noonan group, it's actually related. As you can see here, 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 and here, that this is like a significant connection between how the child is functioning and their brain volume, the striatal volume. And it happens for the Noonan, it does not happen for the typically developing kids. So this is something unique, that we are able to show the connection, not only that the brain is affected, that it's also connected to the behavior. And I'm happy to answer, I'm gonna state that in a very kind of general terms, but and I'm happy to answer a question if someone is interested. We had an ability to also measure how the genes, the PTPN11 is expressed, the gene that causes Noonan syndrome is expressed in the brain, and to correlate that with a measure of the surface area of the brain. And as you can see in general, there's a correlation between how much the gene is expressed in the brain and how much it affects the surface area. So it's another evidence for us that mutation in this gene will cause differences in surface area. And I'm happy to answer more questions. I just don't want to make it too complicated. And now we're expanding, but describing the last section of the talk and the last section of the findings. And this is our first attempt, and I hope first of many, to not only focus on Noonan syndrome, but also start to look on other RASopathies, because we both Beth emphasized that, and I'm emphasizing that because RASopathy is a collection and very connected biologically, a very efficient way to discover new things is to expand to other genes and learn how they function and how they affect the brain. And we do know that, since I told you that the RAS-MAPK pathway activates, the mutation activates the pathway, in general, you think, okay, if everything is activating the pathway, the effects in the brain across this syndrome should be similar. But we still know that they're not the same syndromes, right? They're different. A child with NF1 has different issues compared to a child with Noonan syndrome, but some of the issue overlap, some of the issues are unique. Similarly to that, we wanted to test whether the brain is similar or different. And our assumption was that some of the things will be similar, but some of the things will be different. And we assumed that because we already read previous studies that were done that are outlined here. Just for an example, kids with Noonan syndrome have a decreased total gray and white matter volume, meaning that they have decreased volume of their brain. And children with NF1 have actually increased volume of their brain. So just out of the bat, we see two significant differences between the syndromes. And what you see here is different measures that we took and different mutation. Let me walk you through that. On the first panel, you see when we compare the brain of kids with Noonan syndrome to kids without Noonan syndrome, we find that their surface area, the surface of the brain, is larger on these regions compared to typically developing controls, and smaller in these regions. When we do the same thing for NF1, we discover a different. We discover that the surface area is smaller compared to control here and here. But you can see that the same effects of smaller surface area happened for Noonan syndrome and NF1. So there's some things that are different, but there's some effects that are similar. Then we also looked at how thick is the cortex. And as you can see here, for kids with Noonan syndrome, the cortex was thinner, and for kids with NF1, it was thicker. So we see that there's some differences and some similarities. I'm going to skip those sections, and I want to jump to the subcortical structures that you already know, these deep structures that we're interested in. And this is a different way to view them. But what I want to emphasize is that when we tested kids with Noonan syndrome compared to typically developing controls, most of these structures were smaller. When we tested those structures in NF1 compared to typically developing, most of these structures were larger. But even though they worked in different directions, they worked in different directions in a similar way, meaning that if that was much larger, that was much smaller. If this was less smaller, this was less larger. So we found a very strong correlation between the effect of Noonan syndrome on the volume and the effect of NF1 of the volume, but on different direction. This is the point. So it's affecting in a large amount, but on different directions. This is just to summarize what I just said. But I want to kind of like jump to this, because I think this is going to be important for the population, for you guys who are listening. So the same approach that I showed before, we were able to measure cognitive flexibility. And cognitive flexibility is tightly related to autism spectrum syndromes, because we do know that children with autism have extreme difficulties in moving from one task to another, incorporating new rules and then changing the behavior based on that. And as you can see, the kids with Noonan syndrome and NF1 were significantly lower in their achievement compared to typically developing controls. We were also able to correlate that again with these subcortical structures and show that there's negative correlation between the volumes of the subcortical volumes and the performance. So again, we see this brain behavior correlation that happens for the kids with RASopathies that we do not find in typically developing children. So in conclusion, I hope I convinced you that disease genes in RAS-MAPK pathway are involved in human neuropsychiatric conditions like autism spectrum disorder and ASD. And it's important to study them above and beyond what we need to do for kids with RASopathies. So what I'm trying to say here is that we have the whole justification and the justification to ask for funding to study that, because it's not affecting only kids with RASopathies that are very, very important to us, but we have to kind of like hook ourselves in order to get more funding to do so. So saying that it's important also to the general population gives us a lot of kind of like backing to our ask. And I was what I was hoping to show you is that disease genes in the RAS-MAPK pathway they affect these subcortical structures that are correlating with the behavior that we see, difficulties with attention and inhibition. I did not show you that it also affects the white matter in the brain that helps in connecting between those regions. But I'm happy to talk about it, and that we now are able to expand our studies and looking not only on one gene but on different genes in the RASopathies, and we find some similarities in the effect on the brain and the behavior, and some things that are different and maybe more related to the gene themselves or the protein. And again, to summarize and to say why this work is very important and understanding understanding the effect on the brain is that today we already have, for kids with NF1, we already have children that receiving medication that suppress the RAS-MAPK hyperactivation. There are medications that's called MEK inhibitors, and they're helping in attenuating this hyperactivation of the pathway. This is given not for cognitive and behavioral issues, it's given for tumors that kids with NF1 can suffer from. But if you think about it, and we know that there's effect on the brain, it's something that we definitely need to think about. But if we do give those kids these medications, we need to be able to say whether it also has an effect in the brain, on the brain, and has a positive effect in the brain, making the brain less affected. And again, we actively recruiting participants, and you can just scan this QR code and it will take you to our questionnaire. And lastly, I want to say huge thank you for the RASopathy network that's been supportive of my work for many years now, I think 5 years now. And thank you for the invitation, and to our funders, but mostly for our group of people that work and focusing only on RASopathies in the past 4 years. So thank you.
Thank you so much, Dr. Green and Naomi, for giving us that really interesting talk. I think we know our brains are complicated, but you helped us understand sort of a little bit more about how they have structure and how those structures can manifest certain behaviors. And I have to say, as the parent of a child with Noonan syndrome, I've definitely experienced some of those behaviors in terms of the rigidity and the ADHD. So so I definitely have questions that I can follow up with, and I hope some other folks do too. Okay. So I guess one thing that's maybe a quicker question to answer than the other one was, are these structures in the brain already formed and set by the time a child is born? Or, and so in thinking about future potential intervention, would those things be able to be modified, or are they developing still throughout the childhood, and that we could have behavioral modifications that would help with these?
Yeah, it's a great question. And the answer, as everything in the brain, is complex, but I'm gonna kind of divide it to two things. One is that a lot of the brain development happens in utero. So I do believe, and it's only a belief because no one tested that some of the effects are already there when the child is born. However, the period of development throughout the age of 25 years is a tremendous one for the human brain, and we are going from being really helpless creatures to creatures that can hopefully function in the world independently. And it happens through, we are the longest one, right? So there's a lot of development and reformation that happens after birth. So I would say that some of it is already there, but there's definitely so much plasticity, especially in the younger ages, but not only. We do know there's a lot of plasticity even after that. That there's definitely room for intervention. I think the very important question in what you said, I just want to kind of like emphasize that, is that it's probably very important when you intervene. In terms of like, do you intervene at 5? Do you intervene when the child is 15? Because and that we will know from longitudinal studies. Like if we learn the same patient, the same child,
following people over time. Yes.
Yes. So that's all right, because what the data I showed you is like taking a group, and I can't really answer those questions. So that's definitely going to be our next step. But overall, the answer is yes. I do believe that there's a lot of intervention that can change the course of development.
Yes, we know actually from early childhood development that children from 0 to 3 and even a little further are making 1 million neuronal connections per section. And then there goes a pruning process, and then it all happens at a lower rate during the adolescent periods. So that makes a lot of sense, and be very interesting to find out when that window is for our children.
Yeah. Just for an example, like even there's a study that scanned the brain before learning, I think 10 lessons in piano and after, and you see differences. So there's, as long as you know what you're doing, you can definitely change. So I think.
I do have one more question, if the audience will allow. So I'm curious about the finding that the children with RASopathies have a harder time making and keeping friends. This is something that we've experienced as well. Can you break that down a little bit more in terms of, what's sort of underlying that? Because that's pretty broad. So is it that they are not willing to approach people, or is it that they form connections but can't friendships but can't keep them? Is it because they have a tendency to be either hyperactive or have problems with inhibition, so they're not letting the other person talk? Or do you have any insight into what that really, the basis for that is?
Yeah. So I also went through a process with that, because at the beginning, when I started to work with kids with RASopathies, I thought it's more of the latter. I thought it's mostly the issues with paying attention. And if you just think about it, like if you're a 9-year-old in a group of boys and you need to navigate yourself in this like ping pong of conversations, and keep your attention and keep, follow the conversation and be funny this is really difficult task. So I thought it's mostly the attention component. But the more I got to know the kids and test and see kids in younger ages, and also from the data that I just showed, I think there's a genuine difficulties in the more core symptoms of social cognition, in the sense that it's not only the attention, but it's also the challenge in understanding, for example, facial or affect expression. I don't think it's very gross, meaning that it's not that a child cannot say if someone is happy or not happy, or sad or not sad, but understanding the nuance of this subtle facial expression that kind of like signal to other kids, hey, I'm not interested in talking with you. This is something that a child with RASopathy might miss. And that gets them into trouble, because they might, approach when they're not kind of like not received well, and they can't figure it out. So I do think that it's kind of like both components. If we're thinking about interventions, the earlier the better. And I have now a new dream about, like, we have a VR room, virtual. So like thinking that, I always think about kids with developmental disabilities is that they just need more practice. Like, it's not that they can't, you just need to do it again and again, many times, until these brain connections are formed. So I think that virtual reality would be amazing for that, because you can practice again and again. So this is one of the, so I hope it answered your question.
Yes,
Yeah.
very interesting. Thank you.
I had a question. I noticed the list of autism genes with autism likelihoods, and I saw HRAS on there, and that's Costello syndrome. And we also know that Marnie Axelrad wrote a publication that children who were with Costello diagnosed with autism tended to not have those autistic traits by 5 years old. So then, is it really autism? So I'm really curious about that. We can dig into that, I guess. That really leads me to the question of, so you've looked into Noonan and NF1 and seeing even almost a divergence. I mean, the fact that they're almost mirror divergences also still means they're all kind of interconnected.
Yes.
How about the other RASopathies? How might this have implications for the other RASopathies?
Yeah. I think there's a lot of implications. I do think that in Costello and CFC, the challenge in studying them is the rarity, the fact that they're more rare. But I am, like what I mentioned at the beginning, is that now at the lab, I am taking this approach of like expanding, at least within Noonan syndrome, and hopefully at the later stage, just to study it as RASopathy. I think gonna be, but you have to build a foundation to convince the founders that it's that you can actually after you conduct the study, you can actually answer some questions. So this is challenging, because it's not because like we're able to handle, I think it's more challenging to scan a child with CFC or Costello, but I think we can handle that. The challenge is to have enough kids and have enough like to form a group. This is what is challenging. And for what you asked before about autism, I think autism is a collection of symptoms. So I think like actually just the phenotype of kids with Costello this is the phenotype. Like you don't need to look if they're like, this is the phenotype, this is what's important. And we're trying to fit them into these boxes of ASD and ASDHD, but it's actually how the phenotype is. So I'm not very kind of like hooked, like not very married to this definition. Also, when I treat them, it's just, based on the challenge. Yeah. I think maybe we could open to questions if someone has, because I think people are getting...
Yes. All right. Well, thank you very much. And now I'm going to stop recording. Thank you so much.